Hello students, welcome back to my channel. In this video, I am going to give you objective type of questions from the topic applications of forensics such as PCR and DNA fingerprinting. So let's see the first category of questions such as multiple choice questions. Let's see the first question. What are the components of PCR? Options are DNA, polymerase, reactor. So, second option is DNA, RNA, tag polymerase. Third option is DNA template, tag polymerase, buffer, DNTPS, primers. Fourth option is none of the above. For this, the correct answer is DNA template, tag polymerase, buffer, DNTPS, primers. These are all required to do PCR technique. Let's see the next question. What are the uses of PCR? Options are useful in forensic science and medicine, in research and genetics, amplification of target gene, all of the above. Yes, so these are all the uses of PCR. Let's see the next question. Which of the following technique is associated with a DNA fingerprinting to amplify specific DNA sequence for the analysis? So options are PCR, gel electrophoresis, western blotting, southern blotting. Okay, so the correct answer is PCR, by which you can multiply your DNA, you can amplify the DNA, so whatever you require. Let's see the next question. In the first step of PCR, the two strands are separated by high temperatures. That process is called as. Options are denaturation, annealing, extension, none of the above. So the separation of two strands is the first step of PCR. So that process is called as denaturation. Let's see the next question. So abbreviation of PCR stands for. Let's see the options. Polymerase chain reaction, reactor, polymerization chain reactor, polymerase chain reaction, polyhedral chain reaction. For this, the correct answer is PCR stands for polymerase chain reaction. Let's see the next question. So, who is the father of DNA fingerprinting? Options are James Watson, Francis Crick, Alec Jeffrey, Rosalind Frank. For this, the correct answer is Alec Jeffrey. He is the father of DNA fingerprinting. Let's see the next question. DNA fingerprinting is beneficial in resolving paternity disputes. How? Yes, options are by analyzing the blood type of individuals involved, by assessing the physical resemblance between the children allied father, by conducting behavioral analysis, by comparing DNA profiles of child mother alleged father. So, for this, the correct answer is last one is the correct answer. Let's see the next question. Standard PCR plates have how many wells? Options are 90, 96, 97, 98. Yes. The correct answer is 96. Let's see the next question. Short tandem repeats STRS particularly used in DNA profiling. Why? They are options are they are used in each individual. They are light mutable, mutatable. They are found only in cooling region. They are resistant to degradation. Yes, the correct answer is they are resistant to degradation is the correct answer. Let's see the next question. What is the use of tag polymerase in PCR? Options are thermostable, cheap, easily available, has simple structure, hydrophilic enzyme. Yes, tag polymerase is thermostable. That means it can tolerate high temperatures. Next question. What is the purpose of using gel electrophoresis in DNA fingerprinting? Options are to amplify DNA sequence, to cut DNA into small fragments, to insert DNA into a host organism, to separate DNA fragments based on their size. So, for this, the correct answer is to separate DNA fragments based on their size is done with the electrophoresis. Let's see the next question. How DNA fingerprinting be utilized to identify a suspect in criminal investigation? Options are by analyzing the blood type of the suspect, by conducting hair analysis of the suspect, by assessing the height weight of the suspect, by comparing DNA sample from the criminal scene with DNA from the suspect. So for this the correct answer is by comparing DNA sample from the criminal scene with DNA from the suspect. Let's see the next category of questions. They are belonging to fill in the blank type. Let's see the first one. The first step in PCR is called dash, where the double stranded DNA heated so to separate the strands. So this is the first step. There the DNA to the double helical DNA is subjected to high temperatures to enhance to ensures denaturation. Okay, the separation of two strands is called as denaturation. Let's see the next question. The enzyme used to synthesize new DNA strands in PCR is called, yes, here we are going to use the Taq polymerase. So that synthesizes new DNA strand. PCR requires short sequences of DNA called dash to initiate the replication process. For this, the correct answer is primers. The purpose of PCR is to produce millions of copies of a specific dash sequence from a small sample. The correct answer is from a DNA sequence from a small sample. Let's see the next question. A critical component in PCR that ensures specificity is the design of the dash sequences that flank the target DNA region. Correct answer is primer. Primer sequences that flank the target DNA region. Let's see the next question. DNA fingerprinting involves identifying difference in some specific region DNA sequence called as repetitive DNA. Let's see the next question. PCR was invented by the correct answer is Carey Mullis. Yes. Let's see the next category of questions. They are very short answer type. Let's see the first question. What is DNA fingerprinting? Yes, the answer is DNA fingerprinting is a technique that is used to identify individuals based on their unique DNA patterns. Okay? So it's a technique that is used to identify the individuals by their unique DNA patterns. Let's see the next question. What is the main use of DNA fingerprinting in forensics? So for this the correct answer is, so it is used to identify suspects by comparing their DNA to samples found at the crime scene. Yes. Let's see the next question. What is a DNA probe? The correct answer is DNA probe is a short labeled DNA fragment which is used to detect specific sequences during hybridization. Yes, let's see the next question. How can DNA fingerprinting accurately identify individuals? The correct answer is DNA fingerprinting can accurately identify individuals based on their unique genetic patterns. Okay, so let's see the next question. How does the temperature fluctuate in automatic thermocycler? Automatically, the temperature increases and decreases. That is the answer. Let's see the next category of questions, that is a matching type of questions. Let's see. So here the matching is given. You have to match the list 1 with list 2. So denaturation. So heat is applied to separate two DNA strands. So that process is called as denaturation. Annealing. Temperature is lowered to allow primers to bind to the DNA. That is the correct answer. Extension. DNA polymerase extends the primers to form a new DNA strands. That is the correct answer. Taq polymerase, which is an enzyme used to synthesize new DNA strands, is the correct answer. Primers are the short DNA sequences that bind to the target DNA is the correct answer. So for this the correct answer is A, B, C, D, E is the correct answer. Okay. So let's see the next question. Denaturation. So for this the correct answer is it should done at 94 to 98 degrees centigrade. That's why 96-degree temperature is the correct answer. Annealing, it should done at the temperature 55-degree temperature. Extension of two strands is done at the temperature 72 degree. That's why the correct answer is A and B and C. Let's see the next matching, restriction enzymes. So they can cut the DNA at specific sites. That is the correct answer. Gel electrophoresis. It separates the DNA based on their size in the electric field. VNTRS. So they are the segments of DNA, DNA character to each person. They are called as variable number tandem repeats. So DNA probe, short DNA fragment that is complementary to target DNA. Let's see the next category of questions. VNTRS such as variable number tandem repeats are same in every individual. So you have to tell whether the statement is correct or not. So I'm going to give you the correct answer. So this statement is false. So they are same in every individual is the wrong one. So they are specific for every individual is the correct one. So Taq polymerase is used in PCR because it can withstand high temperatures. So yes, this is the correct statement. PCR is an in vitro technique. Yes, this is also a correct statement. Let's see the next question. DNA fingerprinting is commonly used in forensic investigations to identify suspect or victims. So yes, this is the correct statement. Let's see the next question. Restriction enzymes are used to cut DNA into specific fragments for analysis in DNA fingerprinting. Yes, this is also correct statement. Let's see the next question. PCR is a method used to amplify a specific DNA segment. Yes, this is also a correct statement. Okay. Thank you, students. So now I am going to give you the questions with answers. Now you can take the screenshot. For this, the correct answer is this one. For this, the correct answer is this one, not this. Okay. For this also, the correct answer is this one.